According to conventional medical science, hepatitis C is a viral disease that leads to swelling or inflammation of the liver, leading to jaundice or other issues. Risk factors for hepatitis C are blood contact, long-term kidney dialysis, injection of street drugs, tattoos, if you received blood, or were born with a hepatitis C infected mother. There are two forms: acute and chronic, and we are told that about 1.5% of the population in the United States is infected with the HCV virus. Some symptoms of hepatitis C infection are abdominal pain and swelling, dilated veins in the esophagus or stomach. This can lead to bleeding in those areas, and also nausea, fatigue or even jaundice. Conventional treatments consist of assay antibody tests, but also include undergoing a liver biopsy, as this can show how much damage that has been done to the liver. In 1987, a team of scientists from the CDC and Chiron Corporation went on a hunt for a virus to explain liver disease that they found wasn't being caused by the A or B hepatitis. Researchers began injecting blood from hepatitis patients into chimps. Then they observed that half the animals noted signs of infection of a biological marker of hepatitis called alanine aminotransferase. However, the injected blood did not actually show to cause hepatitis. The researchers began looking in the liver tissue to try to find a virus. They only found a small piece of genetic information encoded in the RNA, presuming it was foreign and viral. They then cloned pieces of genetic information, created genetic sequences and generated proteins that they assumed were coming from the virus's genetic code, and then they created an antibody test based on this. They tested many hepatitis patients who turned out to be positive for this genetic information. Richard Stroman, Professor Emeritus of Molecular and Cell Biology at the University of California, says that this method was unscientific and that the human cell itself could certainly have been the source of the genetic information in the Chiron fishing expedition, and that before jumping on the viral bandwagon, every effort should have been made to completely rule this idea out first. Many other virologists have criticized this as an unscientific approach, including Peter Desuberg, Ph.D., a pioneer in retrovirus research, who is also a recipient of the Outstanding Investigator grant from the National Institute of Health. Desuberg was the first scientist to isolate a cancer gene, and has contributed much to the field of virology. Desuberg is known around the world for rallying scientists to question the HIV/AIDS hypothesis, and is also criticized for standing up for his beliefs, as is the risk of many others who think outside the box. Dr. Joseph Mercola, who runs a very popular natural health website, also has many articles on the subject criticizing the hepatitis virus presumption. I also presume that Kevin Trudeau, Dr. Leonard Coldwell, and the health ranger Mike Adams might question this idea, and many others also speak out publicly about the unscientific methods of many virus hunters. So, are Drs. Mercola and Peter Desuberg quacks, or are they ahead of their time? What we do know is this: to this day no one has been able to isolate this HCV virus, no one has been able to grow it in a culture, nor purify it, nor inject it into an animal to then cause that animal to get hepatitis. These procedures are basic fundamentals of the standards of scientific virology in order to test if a proposed virus is actually infectious. How can we scare patients into taking treatments or living in fear without following a proper scientific method for diagnosis? For those who experience symptoms of hepatitis C, or other diseases, it is recommended to first check to see if you have deficiency in these key 4 areas: minerals, iron profile, amino acids, and B vitamins. Natural minerals like calcium, magnesium, zinc, chromium, selenium, and the proper foods and supplements taken daily can be a powerful approach for those who are deficient or healthy.